Jonathan Villena-Vargas. He's assistant professor of cardiac surgery here at Weill Cornell. So he's going to talk to us about a third cellular source for adaptive T cell therapy in solid tumors. Jonathan?
Thank you, Joe. You can probably tell I'm a surgeon, and I'm a thoracic surgeon, and I've been dealing with lung cancer basically throughout my career, and that's what got me started on this. Just to quickly go over the major teams. So like Jeff said, I'm an assistant professor here. My laboratory studies the benign draining lymph node in lung cancer, which is important. I'm not sure how many of everybody's in the cancer space, but solid tumors almost always metastasize to the lymph nodes first. So one of the original kind of tenets of oncologic surgery developed by William Halsted over 100 years ago in mastectomies is that the solid tumor goes to the lymph node. And that's why it's so important for staging. So in almost every single solid tumor, you remove a lymph node in order to look for staging, right? And what's interesting about the lymph node is that almost all surgeons, and pathologists, if they are benign, meaning that there's no cancer there, they just throw them away, right? They serve no purpose. Now, recently, the addition of immunotherapy such as PD-1 and checkpoint blockade and CTLA-4 and has really revolutionized how we treat most cancers, almost every cancer. And what we are finding in the lab and others have also found is that this benign draining lymph node is actually a center for immunity, right? That's where the T cells get educated. That's where it actually understands the solid tumor, even if the cancer is not there. And that interplay is the reason that the solid tumor goes to the lymph node first, but it's also the first line of defense. So my entire lab is focused on this benign draining lymph node, specifically in lung cancer. And now we're expanding it. So this is my kind of take on this. One of my co-founders here, who I've known since I was across the street working on CAR T cell therapy, he is a basically cell engineering expert. He has multiple high impact journals. He's developed multiple different strategies on how to make a cell better. Uh, if you guys are into the cell therapy space in solid tumors, you'll know that cellular therapy is amazing for liquid tumors or lymphomas, leukemias. There's been 7 FDA approvals in the last 20 years. And they work incredibly well. They're actually moving towards first line therapy. The problem with solid tumors is that unlike lymphoma, unlike leukemias, which have one target, the solid tumor is heterogeneous and it has multiple mutations. So even if you attack one thing, everything else grows out. So that's why CAR T cell therapy has had a difficult time in basically dealing with almost all solid tumors. Now, the recent, and I say recent because it's recent FDA approval, which happened in 2024, but this technology actually came out before CAR T cells, is TIL therapy, developed in Rosenberg's lab at the NCI, in which you actually take the tumor itself, you take the cells from the tumor that recognize the tumor, you grow them out, and you give them back to the patient. And the reason that this got FDA approval, and it's really the only FDA-approved therapy that works in solid tumors, is because those T cells recognize multiple tumor antigens. They're able to recognize multiple of them. So there's no such thing as antigen escape, or much, much less than CAR T cell strategy. So that strategy works. But the reason that, this actually was developed in the 80s and it took until 2024 to reach FDA approval is because it's incredibly cumbersome. You need to take the patient to surgery. You need to grow these cells out, it's an open procedure, meaning that they literally have to cut the tumor open in a big, expensive, sterile room. They have to wait till those cells grow out, which are difficult to work with because of by definition, it's a failed immune response inside the tumor, right? Or else the tumor wouldn't work, or else, we wouldn't have cancer. But it takes 6 weeks to do that. And then importantly, because these cells don't work well from the TILs, they're kind of exhausted. You need concurrent dosing of IL-2. So after the 6 weeks, the patient has to come in, get IV infusions of IL-2, which is very toxic. So up to almost a third or 40% of these patients will actually never receive the therapy even after surgery because the long turnaround, right, 5 to 6 weeks, is the usual. We do this here on patients, so I know firsthand. And they might progress, right? These are patients who already have metastatic disease. Sometimes you take the tumor out, there's not enough cells to grow, so it's unreliable. And then also, IL-2 can be very toxic for the patient, so they don't get all the doses or have some sort of adverse events that prevents them from getting the therapy. So what I'm saying here is that the mechanism of these multiple antigen recognizing T cells coming from the patient's own body is there. It's been there for a while. It's just the execution that needs to be better done. So what's the market opportunity? I think the most important part of this slide is right here. 90% of all cancer deaths in the world are due to solid tumors or metastatic solid tumors, right? So blood cancers that are, CAR T cell therapy market is 10%. We're talking about 90% of all cancers. It's, 1.8 or 2 million deaths a year worldwide. So there's definitely a place to do this. Another important thing is that depending on the cancer, but the majority of the top three cancers, such as melanoma, lung cancer, colorectal cancer, a lot of these cancers are getting immune checkpoint blockade, which we all know. And TIL therapy works on patients that fail immune checkpoint blockade. So it's really important, especially in lung cancer, in which 85% of the patients will get immune checkpoint blockade, but it'll have about a 10 to 15% response. So 85% of those patients will eventually relapse and will need other treatments. So the key insight for this technology is that we find the exact same multiple antigen recognizing cells in the benign draining lymph node. And the reason that's a critical insight is because these lymph nodes have already been mapped. Every thoracic surgeon in the country knows how to identify them and remove them because we do that for staging. But more importantly, they have consistent yields. They're healthier cells. The tumor hasn't exhausted them. So they recognize the antigens, but yet they don't have all that cumbersome that comes from the immunosuppression of the actual tumor environment. So our initial studies look at these cells and say, it takes 1 to 2 weeks to expand these cells as opposed to 5 to 8 weeks. You can do a closed system manufacturing, which you actually don't have to open the tumor and cut it open and expand them for a long time. You can actually do a closed system manufacturing that you'll do in a couple of weeks. That makes it much more scalable. This can be done in a non-sterile room or even a hospital with a blood bank, which every hospital has. And importantly, we also show efficient CAR T cell transduction. So cell engineering on top of these cells, which is the next key step, which they're actually doing in TIL therapy now, is much more efficient in these cells because, again, they're healthier. TIL cells are very difficult to work with. Engineering them is even more difficult. So we're already a step ahead of the game. And the important thing about that is that it's open to multiple CAR T cell strategies that are already out there for solid tumors that have failed because they use the wrong subsets. They don't have that added advantage of the multiple antigens. So again, I do this every day. So it's a straightforward harvest. All these lymph nodes are mapped out. We do this robotically nowadays. It takes about 30 minutes. The patients go home the next day, and every thoracic surgeon knows how to do this. On the last 62 patients that we looked at, we removed average of 20 lymph nodes. That's an average surgery for oncology. So you have a small lung cancer, that's what we remove. 10 lymph nodes. So even if you just take 10, it equals to about 4 billion T cells, which is starkly different from what they get from TIL therapy, which is in the tens to hundreds of millions, right? So you can already see that there's a lot of things that you can do with these. So what's the advantage, right? So right now, like I said, there's 2 different cell sources, one being from the tumor itself or TIL, and the other one being CARs, which is another approach that people are doing for solid tumors. So the T cell phenotype, we find that it's stem-like. And this is published not just by our group, but by multiple groups. There's these special memory cells that live in the lymph node. That's how they take care of long-term immunity. Unlike TIL therapy, which is exhausted, and CAR T cells, which come from the peripheral blood, so they've never seen antigen. Manufacturing time is a couple of weeks. They recognize multiple tumor antigens. CAR T cell is efficient, and other engineering strategies are very efficient. Importantly, we can engineer these cells without IL-2. So our idea is that the patient themselves are not going to need concurrent toxic IL-2 at the time of infusion. And it's very consistent source. We've already shown in our preclinical studies efficacy. We've already shown that transducing these cells in human lymph nodes with patients with lung cancer, this works. These cells work. They expand, they're viable. We've done freeze thaw with them. They're basically as usable as leukapheresis, or what they do for blood cancers, for CAR T cells. Their phenotypes are preserved even after manufacturing. We're optimizing and have a protocol already for closed system manufacturing. So, again, we're starting with a healthier T cell that really streamlines things for us.
John, we have about 2 minutes.
Thank you. So, what are we now? We call this a platform because, like I said, we are doing two things. At first, we're looking at bulk LNTs, just like bulk Ts are TILs, meaning that they don't need engineering at first. That's just for initial FDA approval. But then we're also doing a platform in which we're using multiple engineering strategies that use the same source. So right now we're looking for some seed funding. We're going to do IND studies. We already talked to the head of our GMP facility here. We already have everything protocolized, and we just need this to move forward. So it's in the initial set. This is protected by a patent, which is done here at Cornell, and we have a couple of other ones in the pipeline as well, mostly with the manufacturing and closed systems. Like I said, the mechanism has already been validated by Iovance. This FDA approval and this sort of surgical T cell approach is there. We've exposed the gap why Iovance and TIL therapy may struggle. It's lack of scalability in the manufacturing aspect of it. And like I said, it's a platform on which we can try multiple engineering strategies. So the ask, like I said, $500K for IND enabling studies, which we think we can do in the next 18 to 24 months. Thank you. Happy to take any questions.
Okay, thank you. You can open it up for probably one, maybe two quick questions.
So I do have one quick question. I was actually at Advaxis, which was a Listeria-based immuno-oncology immunotherapy company. And one question that I do have is if you can just kind of walk me through the process of actual manufacturing at a very high level. I think that would be very beneficial, because when I was looking at that, it is the tumors are heterogeneous, you are correct. And if we have a multiple neoantigens, how are you actually planning to construct them, and how are you actually planning to deploy them in the patient? If you can just walk me through that.
Sure. So the T cells themselves are there in the patient. They already recognize the tumor antigens, right? Just like TILs. And the key to the manufacturing is that they don't need open dissociation or filtration. We actually have a device where a surgeon can get it in the operating room, put it into the device. That actually makes it very easily into a single cell suspension that will then go into, one of those leukapheresis bags, like
Right.
when you transfer anything. And that'll go, and we have a collaboration with Scale Ready, which is the G-Rex, basically the cell manufacturing platform people. And then goes from there to this other closed system where it grows them out for a couple of weeks. So that protocol is there. And then the infusion is much like any cell therapy infusion, in which you have to treat the patients first with cyclophosphamide or lymphodepleting therapies, and then you infuse the cells.
And then what is your earlier data in instead of human, right? Like I see that you are planning to go into IND in 18 to 24 months. what has been your preclinical data look like?
So the preclinical data, we've shown that we've been able to transduce three different strategies. In the mouse models, we've used multiple patient strategies. So we use actual patient lymph node T cells from lung cancer patients, and those have been shown to be usable probably a dozen times. So we worked out the manufacturing. The manufacturing also has been worked out, so it's completely closed. We don't use any beads, no debeading. Everything is from the outset, we wanted to streamline this thing in order to make it scalable and easy. And the efficacy is there when we do head-to-head TILs versus LNTs. LNTs always win out. They're just a healthier cell.
Okay. Well, thanks so much, John. So